This is a guide video for Worksheet 1, just to walk you through the concepts and explain some of the things, some of which we got in class also. So steps 1 and 2 are just guides to help you walk you through opening the file. Use the .mas one unless you're on a Mac, or if you have an issue, you can put in this .fas, which is just a text document but has all of the DNA sequence. Once you get it open, you should see this alignment window, which is a sub window within the Mega program. So let's just walk through the worksheet starting in number 3. So this is a small data set. There are 6 different genes in it, 5 that we know the species for, and then 1 that we don't. Okay, so just to do it, and also to point out there are 2 little bits of data here that also give us a little bit of a clue. These are unaligned sequences. We'll talk about that in a minute, but it just means they're raw sequences these were downloaded from NCBI, the online database, and that these are homologous nucleotide sequences, or you could say homologous genes in this case. So these are genes. Homologous means they come from the same ancestral gene. So if we were to find the common ancestor of all 6 of these species, we would see this same gene in that species, and so any differences between these different sequences are mutations that have occurred and been selected for, or been selected by, or have natural selection or genetic drift that have made them part of the genetic background of each of these species. And so that's why there would be differences. And we're going to come back to the alignment in a little bit. Okay, so number 4, which gene are they from? Now, right away, and part of this is just to get you comfortable working with DNA sequences and with this piece of software. Right away, you notice that these genes are already labeled with both information about the organism and then the gene name. So if we trust the gene names, it's an 18S ribosomal RNA gene. However, one of the things you can do, depending on the data set, and with the .mas file you can do this, you can right-click and then go back to the original page from which the sequence was downloaded. So I now have additional information about the sequence, about where it was published originally, what the researchers were, and then information about the gene. It's in the title, right? So you can see it in multiple places. It's an 18S ribosomal RNA gene. Okay, so that answers question number 4. You can just put that here. You know, what are these sequences are from an 18S ribosomal gene, or you can say ribosomal RNA, right? So is this gene mitochondrial or nuclear? Now, if you don't know off the top of your head, there are 2 ribosomal genes that are in the mitochondrial genome, and there are 2 of them that are in the nuclear genome. One codes for a large subunit and one codes for a small subunit, and they're slightly different. Mitochondria have their own ribosomes. This 18S is a nuclear gene. Okay, so that is important in some context, so it's kind of good to know about these. If you didn't know that, you might have to do a little bit of a background work, Google it, figure it out. But most of a human genome is nuclear, meaning it's found in the nucleus. There's a small amount in a circular form, so double-stranded DNA still, but in a round circle instead of in a chromosome. That is part of your mitochondrial genome. Okay, now, is this gene protein coding? Again, this is a little bit of background. Most genes are protein coding. This is one of the rare exceptions. It's a ribosomal gene. So it's transcribed into single-stranded RNA. It folds into a 3-dimensional shape, and then that makes up one of the subunits of these functional ribosomes. And ribosomes, of course, help to facilitate DNA translation. So they are part of the translation process, but this gene itself is not translated. Okay? So it's not protein coding, but then kind of reviewing our definition of a gene. How can we call it a gene if it's not protein coding? Well, remember our definition of a gene. So you could, if you wanted to here, put non-protein coding. And then reminding us ourselves, the definition of a gene is a piece of DNA that's transcribed. And then if you wanted to, you would include that it has some sort of an impact, right? The transcription is either, in this case, used to do something, or is then translated, processed, and translated into a protein that does something. So you could put a piece of DNA that is transcribed and has an impact, right? some sort of function on the organism. That's our definition of a gene. Okay, so these can be genes even though they're not protein coding, and that has some ramifications in how we might analyze it, look at mutations and things like that. Okay, so what organism does the mystery sequence come from? So we've got a mystery sequence that's unlabeled here. Now, even if you just looked at the sequence, you could probably infer that it is an 18S gene, because notice it is quite similar to all of these other ones with a few differences. However, we have a tool. So I'm going to introduce you, if you're not familiar with it, to what's called a blast search. You could copy and paste this onto the website that does a blast search, or in Mega, there are links. So you can do here and do a blast search once you've highlighted a sequence, or there's a little shortcut right there. So that shortcut will open a new window and automatically load any sequence you have highlighted into the search window. If you didn't highlight a sequence, it will just open up a blank window, and then you'd have to copy and paste your sequence here. But notice I now have this mystery DNA sequence preloaded. We'll leave everything on default and hit this button, which begins the blast search. You can think of this as like a Google search for genes. So it is now searching the entire database for a match to this gene, and it will return any complete 100% matches, and then also a list of other things that are pretty close, maybe close relatives, a gene, a homologous gene from a close relative. Typically, depending on the load on the servers, this will take anywhere from a few seconds to a minute, minute and a half, sometimes a little longer if it's very busy, like on weekday afternoons. So it's nearly done. I'm going to let it refresh one more time. It refreshes automatically. It just waits a little longer for each successive refresh. So let's wait for one more refresh. There we go. And if at any point this stops refreshing, you can just refresh the window there, reload that page yourself, and if it's done, it will pop up. Okay, it is in the afternoon here where I'm doing this, so let's see if it will work after this one more. It usually doesn't take more than a minute, minute and a half. Okay, we'll let that go and we'll come back to it. Okay, so we're searching to give a name to this DNA sequence. Now, one thing I want to point out while this is going, let's just take a quick look at this entire length. So you can do that by grabbing this little bar and scanning across the gene. And what you'll notice is at the beginning, they're all somewhat similar. There are some differences, and they seem to unite certain groups of species seem to be quite similar, and others are a little different. But then as we scan down, you'll notice they're becoming quite different. And even though we can kind of see the patterns, they're not really lined up well. And so although there are still similarities, by the time we get near the end of the gene, only very closely related species are still showing high levels of similarity. And even those, after a while, might become a little bit shifted and off. So notice, even at the end, we see that some sequences are longer than others, right? So I want you to maybe just refresh your memory. When I see a data set like this, and I see everything quite similar, but then a difference, that represents a substitution mutation somewhere in the past. And if two species share it, it's likely that was a substitution somewhere in their ancestor, and they both inherited it. But these other ones didn't have that ancestor, so that lineage diverged before the mutation. And the reason that these two have a T there or a C here or these sequences over here is because they've accumulated differences after they diverged from the rest of these species. But if we see a length difference, that is representative of an insertion deletion or probably multiple insertion deletion events. And so if we are going to compare homologous positions in the gene, we need to perform an alignment. Okay, so let's check and see if our search here is done. I can close that window. Let me open up the other one. Okay, so here is our search window. It's still refreshing, so it's a very busy time right now, I guess. We've gone 3 minutes now. We'll let it go one more time. Let's move on, and then we'll come back to this just for time's sake, okay? So the next step, we're going to try and take care of this difference that has been noted because of insertion and deletion events. And to do that, we need to perform a process called an alignment. So now I'm going to highlight, you can click the top one and hold down Shift, and while you're holding down Shift, click the bottom one. That will select all your sequences. You can also just do Control A. Once all our sequences are selected, we're going to choose one of the alignment algorithms. There are a couple that this program allows us to use. You can also do it by alignment here, and then there's a drop-down menu. But this little shortcut is for one of those called muscle. We're going to align DNA because we don't have codons, it's non-protein coding. Leave everything at the defaults, and it should, within just a couple of seconds, complete an alignment. And now when we scan through these genes, you'll notice that even though there are areas where there are insertion deletions, so this little gap or dash represents an insertion deletion event. And at the end, it's very conserved and everything lines up well. In the middle parts, there are parts that line up really well with a few substitutions. And we have variable areas. So parts of this gene are much more variable and seem to be tolerant of insertion deletion mutations, and others are very, very conserved. Now, when you look at this doesn't mean there haven't been mutations that have been occurring. It probably more likely means that when mutations occur, they're selected against in this area and not allowed to be maintained in those species. But here, mutations that build up gaps in space are a little bit more flexible, and so more often they're kept by natural selection or genetic drift. So it's a little bit more of a flexible place. Mutations tend to happen randomly, but then natural selection especially tends to make distinct patterns that we could use to analyze different parts of this gene. Again, so here's our beginning, right? Fairly conserved, few differences, variable regions, really variable region, right? And then it's conserved again. So now we have completed our alignment. All right, so here are the results. Took about 4 minutes. But we have the very first sequence here that is a hit, 100% coverage and 100% identity. And then notice we have some close matches, but we found the gene itself here. So we know what gene it is and what species it came from. So here is the genus Necrophila americana. You can click on that and it'll take you to kind of a summary and even a link to the actual page if you want to look closer at it. This just shows you how it matches up, 100% identical, so that's kind of a given. But here's the link to this specific gene sequence in this species. There's a common name, so it's the American carrion beetle. And sure enough, it's the 18S ribosomal RNA gene, same gene, which we kind of already knew based on how similar it was. So the worksheet asks you then to put in the name here. So we're going to put in Necrophila americana. And it doesn't ask for this, but we could also put in the gene name, right? It's an 18S ribosomal gene, just like these others. So there we go. So now we have identified our mystery sequence. We've aligned all the sequences together. The last thing we need to do is just answer some follow-up questions, which we've kind of already covered, but let's make sure they're clear for you. Okay. So the organism, we've already labeled that mystery sequence with the genus and species. We've performed our DNA alignment. We use that muscle algorithm. And now we're answering, are there any gaps in our alignment, right? So a gap is represented, instead of having an A, a T, or a C, or a G, we just have a dash, right? And so that gap represents a insertion deletion event. So certainly, yep, there are gaps in our alignment, and those gaps represent insertion deletion mutations. And we treat them similarly because they have a similar impact. And in reality, if I see this here, you don't know whether it was a deletion of that position in these two species, or more probably their ancestor, or maybe it was an insertion of a G in these other four, or an ancestor of those four. So we would need to maybe do some additional work to detect whether it is an insertion or deletion, but for the alignment purposes, we treat them as if they are equivalent. So that's one reason why we call them indels or insertion deletions and often use those terms together. All right, so you're going to get future opportunities to work with Mega, but that's all you need to do for this worksheet. So the last thing is just a little instruction. Save your DNA alignment. So we're going to go ahead and save this one. Okay, save it somewhere where you know you can leave it in that same format. And then we're going to save our worksheet with the answers in it. And then all you do is go to the assignment, attach both of those files, and submit this as the completion of your worksheet number 1.